Armando Hasurugan, biology and medicine videos. Please make sure to subscribe. Join the forum and group for the latest videos. Please visit Facebook, Armando Hasurugan. You can also like here. Please ask questions, answer questions, and post some interesting things, including your artworks. In this video, we're going to look at a human metabolic pathways, and we will follow the fate of one glucose molecule and some oxygen we breathe, and what the whole metabolic process from these two substances will yield for us. This process is known as cellular respiration. So we begin with glucose from outside the cell, taken in by a GLUT transporter. So now the glucose is in the cytoplasm of this particular cell. For now, this cell can be any cell. Let's just say it's a liver cell, a hepatocyte. Before we continue with this map, let's have a look at what we're looking at in this first part of the video. Now, cellular respiration can be written as an equation, an overall equation. So overall, remember, which is one glucose molecule plus 6 oxygens will yield 6 carbon dioxide, 6 water, plus some energy in the forms of ATP. Now remember, we are inside the cell. And inside the cell, we have mitochondria, the ATP producing organelle. Now, and of course, outside the mitochondria is the cytoplasm of the cell. The mitochondria has two membranes, the outer membrane and the inner membrane. And between these two membranes is the intermembrane space. The outer membrane of the mitochondria is much more permeable to small substances. So the process of cellular respiration begins with one glucose, let's just say. And this one glucose will essentially make two pyruvate molecules in a process known as glycolysis. The oxygen will come later on. Now, glycolysis also produces two important things as well. It produces 2-nicotinamide adenine dinucleotide, or NADH, and it also produces a net of 2 ATPs. Next, what happens is that the two pyruvate molecules produced through glycolysis will enter the mitochondria. And inside the mitochondrial matrix, the two pyruvate molecules will convert to 2 acyl-CoAs. And this step is known as the preparatory step. And again, this preparatory step also makes an important substance, which is, again, 2-nicotinamide adenine dinucleotide, or 2 NADHs. So let's just stop here for a while with the 2 acyl-CoAs and go back to the metabolism map and look at these two processes, glycolysis and the preparatory step, in a lot more detail. So first, we're looking at glycolysis from glucose. So first of all, glucose will convert to glucose 6-phosphate with the enzyme hexokinase. And the phosphate is obtained from ATP. Next, glucose 6-phosphate will convert to fructose 6-phosphate with an enzyme known as phosphohexose isomerase. And because glucose 6-phosphate is an isomer of fructose 6-phosphate. The fructose 6-phosphate will then convert to fructose 1,6-bisphosphate with the enzyme phosphofructokinase, and it obtains another phosphate from ATP. Next, glucose 1,6-bisphosphate will then split in half to yield glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate has an aldehyde group, and the dihydroxyacetone phosphate has a ketone group. And the enzyme used is aldolase. This reaction is reversible. Next, dihydroxyacetone phosphate will actually, in glycolysis, convert to glyceraldehyde 3-phosphate with the enzyme triose phosphate isomerase. And so now we actually have 2 glyceraldehyde 3-phosphates, but we'll just write it as 1 for simplicity. But just keep that in mind, we have 2 glyceraldehyde 3-phosphates. So we have, whatever we make, we have 2 of those. Next, glyceraldehyde 3-phosphate will convert to 1,3-bisphosphoglycerate with the enzyme glyceraldehyde 3-phosphate dehydrogenase. The phosphate is obtained from inorganic phosphate, and NAD is reduced to NADH. This reaction is also reversible. 1,3-bisphosphoglycerate will then convert to 3-phosphoglycerate, or dephosphorylate to 3-phosphoglycerate with the enzyme phosphoglycerate kinase. and the phosphate is taken by ADP to form ATP. This reaction is reversible. 3-phosphoglycerate will then move its phosphate to carbon number 2 to form 2-phosphoglycerate. And this is done by the enzyme phosphoglycerate mutase. And this reaction is reversible. 2-phosphoglycerate will then dehydrate to form phosphoenolpyruvate. The enzyme used in this reaction is enolase, and this reaction is also reversible. Phosphoenolpyruvate will then finally convert to pyruvate through the enzyme pyruvate kinase, and the phosphate is removed by ADP to form ATP. So we actually have the final product is 2 pyruvate molecules because we had 2 glyceraldehyde 3-phosphates. And so if we look through the whole process, we use 2 ATP, but we make 4 ATPs in total, which gives us a net gain of 2 ATPs from glycolysis. And we also have a net gain of 2 NADHs. And this was through one glucose molecule through glycolysis solely. Another source of glucose is from glycogen, through glycogen degradation. Glycogen can break off its glucose monomers to produce glucose 1-phosphate. And this, many glucose 1-phosphate, and this is done by the enzyme glycogen phosphorylase. And the phosphate is obtained from inorganic phosphates. Now, glucose 1-phosphate can then convert to glucose 6-phosphate with the enzyme phosphoglucomutase. And then this glucose 6-phosphate is then what enters glycolysis to produce a net of 2 ATP and 2 NADHs. Now, going back to our 2 pyruvate molecules, our end product of glycolysis. But remember, we're only going to look at 1 pyruvate for simplicity. So the pyruvate, which is in a cytoplasm currently, will want to go inside the mitochondria. And so it can pass through the outer mitochondrial membrane pretty easily. And then it will need a pyruvate transporter to actually go through the inner membrane. So now the pyruvate is inside the matrix of the mitochondria. The pyruvate will then convert to acetyl-CoA through the enzyme pyruvate dehydrogenase complex with the cofactors thiamine pyrophosphate, lipoate, and flavin adenine dinucleotide. And also in this process, a CoA group is added and NAD is reduced to NADH. Also, a carbon dioxide is produced from this reaction. So now let's stop there and go back to our very early diagram. So we have acetyl-CoAs now. We actually have 2 acetyl-CoAs. And so these 2 acetyl-CoAs will then enter what's called the citric acid cycle, also known as a Krebs cycle. And in the Krebs cycle, the 2 acetyl-CoAs will produce a few important substances that we must know. First of all, it will produce 6 NADHs and 2 FADHs. And also, the citric acid cycle with the preparatory step will produce a total of 6 carbon dioxides. And the Krebs cycle, the citric acid cycle, will also produce a net of 2 ATP. Let me just emphasize that the preparatory step produces from 2 pyruvates, produces 2 carbon dioxides, and the Krebs cycle produces 4 carbon dioxides. And so this gives us a total of 6 carbon dioxides. So let's go back to the metabolic pathway, the metabolism map, and look at the Krebs cycle, also known as the citric acid cycle, in a lot more detail. So we begin with 1 acetyl-CoA here. We should actually have 2 acetyl-CoAs, but we're just working with 1, remember. So the acetyl-CoA with another molecule known as oxaloacetate will yield citrate through the enzyme citrate synthase. Water is used, and also the CoA group is removed. Next, citrate will convert to isocitrate in a 2-step reaction. First, citrate will dehydrate to cis-aconitate, this is wrong, through the enzyme aconitase. Then cis-aconitate will then hydrate to form isocitrate. The 2 previous reactions were reversible. Next, isocitrate will then decarboxylize to form alpha-ketoglutarate with the enzyme isocitrate dehydrogenase. So decarboxylation will produce carbon dioxide molecule, and also this reaction will produce NADH. We're putting NADH in the middle here because it's important and we'll talk about it later on. Next, alpha ketoglutarate will then again decarboxylize to form succinyl-CoA with the enzyme alpha ketoglutarate dehydrogenase complex. A CoA group is added, and because it's a decarboxylation process, a carboxyl, a carbon dioxide group is removed. Also in this reaction, NADH is produced. Next, succinyl-CoA will convert to succinate through the enzyme succinyl-CoA synthetase. GDP with an inorganic phosphate can get converted to GTP, and also the CoA is removed in this process. This reaction is also reversible. Now the next reaction is important, the conversion of succinate to fumarate through the enzyme succinate dehydrogenase. The interesting about this reaction is that the enzyme succinate dehydrogenase is actually situated within the inner membrane of the mitochondria. So it would be situated here if we have the two mitochondrial membranes. And so succinate will be converted to fumarate through the succinate dehydrogenase. And in this reaction, FAD is reduced to FADH2. This reaction is also reversible. Next, fumarate will get hydrated to form malate through the enzyme fumarase, and this reaction is reversible. Finally, malate will convert to oxaloacetate through the enzyme malate dehydrogenase, and also NADH is produced. Now, because we're only looking at one acetyl-CoA here, we actually have to double everything we produce. So in the Krebs cycle and the preparatory step, so from two pyruvates, which converts to acetyl-CoA and then enters a Krebs cycle, we will produce a net of 2 ATPs, 8 NADHs, 2 FADHs, and also 6 carbon dioxides, which I have not written here. Actually, let me just redo everything again. So from 2 pyruvates to 2 acetyl-CoAs, the preparatory step will produce 2 carbon dioxides and 2 NADHs. And that was for the preparatory step. Now let's look at the Krebs cycle. The Krebs cycle total production will be 2 ATPs, 6 NADH instead of 8, 2 FADHs, and 4 carbon dioxides. So what do these NADHs and FADH2s do? Well, they are known as electron carriers, and we'll talk about them later on. But what we should do is we should go back to our early diagram and tally up, calculate how many NADHs and FADH2s in total we have. And so if they all move inside the mitochondria, we'll have a total of 10 NADHs and 2 FADH2s. And what will happen with them? Well, they will enter what's called oxidative phosphorylation, also known as the electron transport chain. And we'll look at this in the next video. Thank you.